Hello, my name is Erica Lyons, and I'm going to be presenting our poster on literature mining for rare disease phenotype-genotype associations on behalf of myself, Daniel Watson, Jack Collins, Gregory Tawa, Sherry Huggabrook, Elizabeth Ottinger, and Uma Budinuri of the Advanced Biomedical Computational Science Group, Frederick National Laboratory for Cancer Research, and the Division of Preclinical Innovation, National Center for Advancing Translational Sciences. The overall goal of this presentation is to connect with text mining experts because we are actively exploring collaborations and have open positions to support this challenging task. First, the abstract. Although individually uncommon, collectively rare diseases affect 6 to 8% of the world's population. Diagnosing rare diseases through phenotype is a difficult task because symptoms overlap based solely on phenotype. There may be multiple causes for a single disease, and a single cause may be associated with multiple diseases. Genomic investigations are leading to precise molecular-level characterization, allowing for a systematic discovery of therapies that either target a specific disease or, more broadly, multiple related diseases. Currently, text mining techniques have lower accuracy and more coverage gaps than manual curation, but the potential for higher productivity in the future. Recently, there has been some progress in developing text mining applications to tackle this enormous problem. However, there is still a need to assess these text mining applications and integrate the findings with data. Towards these ends, we manually created a rare disease variant dataset which can be used to test and refine a text mining algorithm. We developed a manual curation workflow which incorporated searching for genetic variants on PubMed, listing symptoms and phenotypes associated with each variant, converting cDNA or protein to standardized genetic notation, obtaining annotations from public data sources, and presenting the details in an online interface for future release. Our project aims include summation of pathogenic variant frequency in populations to estimate birth prevalence for each rare disease. We created test datasets of known accuracy, coverage, and genotype-phenotype associations that can be used to validate text mining approaches. Enhanced text mining will significantly decrease the amount of time necessary to gather data to molecularly characterize a rare disease and render it possible to mine rare disease phenotype-genotype associations for the more than 7,000 rare disease genes in a timely fashion. Our methods. Manual curation, harmonization, and presentation methods as shown in Figure 1 are as follows. The curator searched PubMed for the gene name, disease symptoms, and biological pathways of relevance. The curator created a spreadsheet list of all SLC6A8, which is the gene associated with creatine transport deficiency, variants, documenting the symptoms and phenotype associated with each variant. The types of information recorded from the manuscripts are shown in Figure 2. The information collected was assessed for accuracy by comparing to the ClinVar database of genetic variants. 100% of the ClinVar variants noted as having been published had been found by the manuscript curation process. During the harmonization step, the cDNA or protein format of the variant was converted to standardized genetic notation using a custom-developed software. The results were then manually checked and harmonized. Details were used to improve validation accuracy and to improve the software's sensitivity and specificity. In cases where the published variant name did not match the standard notation, the manuscript's text was investigated for context. For example, when a variant's amino acid was referred to as both L and K, context of participation in a leucine zipper led the curator to conclude L. Error-prone abbreviations were noted to include N, Q, D, E, and L. A VIA was employed to obtain annotations from public data sources and analyze the impact. Overlap between the manually curated variants and those in the ClinVar database was assessed. Lastly, the results are to be included on a future released website that will be available to the public to access and visualize the rare disease variants on 2 and 3D protein structures, as shown in Figure 3. Figure 1 shows the tool's workflow for the creation of variants from PubMed, harmonization, and creation of a publicly available 3D variant display. This is our workflow. Figure 2 shows the information that was curated from PubMed manuscripts for the researchers and the clinicians to view in the table on the website. Information collected includes mechanistic details such as protein domain, amino acid conservation, subcellular localization, potential binding partners, activity of the variant as a percentage of the wild-type activity, variant details such as genomic, RNA, cDNA, and amino acid notation, variant type, detection method, and prevalence and incidence if that's known, and the population that it came from. Patient information details include the family, ethnicity, sex and gender, IQ because creatine transport deficiency has an IQ symptom, and whether or not the variant was known to be a de novo mutation. Clinical details collected include the ISMB pathogenicity score, the pathogenicity evidence, disease name, symptoms, test result data, age of onset, and age of diagnosis, including if there was an initial misdiagnosis, and if so, what that was. Figure 3 shows the website that, in the future, will display the 3D protein structure and a sortable list of known genetic variants. Clicking on the variant highlights it on the 3D structure. This will be released in the future. Results. Overall, 190 SLC6A8 variants were curated from PubMed. Of these, 91 were pathogenic. Single nucleotide missenses were the most likely to be properly converted and harmonized, while insertions, deletions, frameshift, and intronic variants are more susceptible to improper conversion during harmonization. The ClinVar database had 278 variants with 50 or fewer base pairs. ClinVar had 51, or 27%, of the total curated variants. ClinVar had 16% of the pathogenic variants curated from PubMed, as shown in Figure 4 below. Figure 4 shows the overlap between these manually curated variants from PubMed and the ClinVar database as of March 2020. Future directions. The curated SLC6A8 dataset can serve as a test dataset of known coverage and accuracy to train text mining algorithms to mine PubMed for rare disease gene variants. The curation process, including initial review, standardization, and validation using context, can improve sensitivity and specificity of such automated algorithms and help expand the extraction of gene variant pathogenicity relationships for the more than 7,000 known rare diseases. We are actively exploring collaborations with text mining experts and have open positions to support this challenging task. Thank you to our funding sources, the National Cancer Institute, the National Center for Advancing Translational Sciences, and the National Institutes of Health.